So now we will not wait. Good evening, all the participants and resource person, Dr. Piyush Agrawal ji. As you all know, this is free webinar series, science dialogue series. Mainly we focused on biological science. And in this series, we have completed more than 100 lectures. And participants are from all over world, mostly from India, but some participants are from outside India also, because in online mode, we cannot restrict and we should also not restrict. And the speakers are also very renowned. In this series, today's speaker, Dr. Piyush Agrawal ji, is very young, but his profile is very, you can say, world. He has got experience from the world renowned institute. Like he did his PhD from ImTech Chandigadh. This is CSIR Institute.
Yes.
And award given by JNU, Jawaharlal Nehru University. And he did his postdoc also from world's top institute, that is National Cancer Institute of National Institute of Health. National Institute of Health, who are postdoc fellow or post PhD, they must have idea. This is top institute in the world, NIH. Generally, NIH fund different institute, but it has also some like a DBT के अंदर जैसे बहुत सारे इंस्टीट्यूट है, उसी तरह NIH के अंदर बहुत सारे इंस्टीट्यूट है, जिसमें कैंसर इंस्टीट्यूट भी बहुत ही वेल नोन है ना? Am I right ना?
Yes, yes.
Yes. So we are fortunate to listen you, and not we means me, also so many participants. Participants are also from graduate student to faculty, scientist, because we don't restrict, anyone can join. So thank you very much for accepting our invitation and sparing your time, sparing your knowledge, vast knowledge. Generally, people get opportunity in offline mode, but here I am making it free of cost for all, so that they can interact with the researchers from well-known researcher and reputed researchers. So this is brief. I will not say more. Ki you have published so many public research paper also in high cited or high impacted journal also. उसमें से साइंटिफिक रिपोर्ट है, सेल में भी मैंने देखा, सेल इंटरेक्शन है. So these are all very high impacted journal. So thank you very much again. We are cordial, means heartily grateful to you for this, Hannah, accepting our invitation. Now platform is yours, and I generally participants, I have not muted any participants, so they can ask question whenever they find difficulties, but generally we prefer after lecture. But if they face some difficulty, they can unmute and speak. Otherwise, you can take.
Okay. Yeah. Thank you, sir. Thank you very much for your kind words. And first of all, I would like to congratulate you for organizing this kind of series, because, in India, organizing any free thing is like very rare. So yeah, but this is like truly benefiting all the candidates, especially the bachelor's and master's students who are not aware much from this field. And the best thing is that you bring a lot of people from all over the world on a single platform, so people have a like broader uh, exposure to different kind of research different speakers are doing. So yeah, I mean, once again, thanks a lot for inviting me and having me here, and congratulations for this wonderful initiative. So yeah, good evening everyone. As sir told, my name is Piyush, and I am basically a computational biologist and bioinformatician. So my research area is in cancer genomics, and other than that, I am also working in the area of antimicrobial resistance, cardiovascular disease, and drug designing. So yeah, feel free to ask me in between if you have any doubt, any question, and let me know if any other sort of issue comes or anything is not clear. I will try to keep my slides and presentation and topic very simple so anyone can understand. But in case you find some technical difficulties or anything is not clear, do let me know. So I'll share my screen with you all. So is the slides visible?
Yes, visible.
Okay, fine. So let me start. Okay. So, as you all know, the title of my talk is Network Based Approaches in Cancer Therapeutics and Biomarker Discovery. So, the motivation for this kind of project comes because if you see, most of you have read papers, and what people do when you have two different type of data, a case and control, that we go for finding differentially expressed genes, and then we do different kind of analysis, enrichment process or whatever. But the problem with DEG based approach or this kind of approach is that it is biased. It do not represents the whole general transcriptional response, and these are not context specific. Hence it leads to false positive results. Okay, so the thing is, network based approach provides a more comprehensive picture compared to differential expression gene based approach, because in network you not only find the whole network which is altered, the key genes, but also the associated genes, because in cell or in body doesn't anything doesn't happen in isolation. The whole network goes on. Whole, proteins are connected with the genes are connected with each other in a network manner. So the global transcriptional change. So when I say transcriptional change, it means gene expression change. So the global transcriptional change, it represents the terminal event and may not be necessarily the key regulator of those changes. For example, if you see, there are many genes which are associated with some kind of transcriptional regulation or the changes in expression, but they are not very much captured when you look for the differentially expressed genes. For example, transcription factors. So if you see the log fold change of those transcription factors, it's very less. And what we generally do when we look for the DEGs, we select top 1000, top, or anything above log expression fold 2 or 1. But what happens sometimes, those transcription factors which are actually related or regulating the whole process, they have a very low log fold change difference in two condition. But at the same time, they are very important in with the whole transcriptional change, which is ultimately leading to a kind of condition, for example cancer or any kind of disorder. So if you see these two papers, they have discussed the drawbacks of this kind of approach, and when you go through it, you will find what I just tell you. But they have not mentioned about the network based approach. They have just shown the limitations of the differential expression approach. So what we have done, so we developed a method called path ext, and in this particular method, what we have tried to do that instead of finding differentially expressed genes, we find differentially expressed pathways. So the input is a knowledge based network. So this is a experimentally validated knowledge based network where you have the data of which gene is interacting with which gene in a network manner. And then we have our omics data. This data could be transcription data, that is gene expression change, or it could be epigenomic data, say DNA methylation data or acetylation data, or it could be proteomic data. So this could be anything. And this is a knowledge based network. So what we do, we take these two input and then we overlap them. So what the method do, that it creates a network with a weight, and then we try to rank the different kind of networks which have been formed from high to low, and then we look for the shortest path. Shortest path means if this is your central gene, then we look what are the genes which are connected to it in a very short distance. So gene miss node and interaction miss means edge. So when I say node, it means gene, and when I say edge, it means interaction. So if you see, if this is your key gene, then it is interacting in the short with this particular gene, this gene, this gene, this gene, but not with this gene. Okay, so what we do, we try to find such kind of closely interacted networks. It means that these network as a whole is working together or is upregulated or downregulated in a given condition. Then what we do from this kind of network, we identify the ones which shows the alteration in disease condition compared to control. So we create this network in control also, and we create this kind of network in case condition also. And then we find that which particular network is differentially, altered in case condition compared to control. It means either this particular network's whole expression is up or down. So then we identify those kind of networks. And what we do to make it statistically that it is not random, okay, we do it multiple times. It could be from 200 to 1000 times, and it is very highly computationally intensive. So because if you do for like 200 times for a single sample, this may take up to 3 to 4 hours, and if you do for like 1000 times, it might take up to 10 hours. So but the thing is, it is more promising, more accurate, more statistically significant, and gives a true actual picture of the disease condition which has been altered. Afterwards, what we do, then we do various kind of permutation based significance estimation. So for every network, whatever, every time we do this network creation and identification, we compute a p value and false discovery rate. False discovery rate tells you that it is corrected for the p value based significance. And afterwards, when the whole permutation is completed, we select the those network which have a q value or fdr less than 0.05. So in the end, what we get list of topnet or altered network. So altered network is also called topnet because it is coming in the top in among all the networks, so we have named it as topnet. So you get the significant altered network in sample specific manner, and then you can analyze that what were the pathways or the processes which were altered in that particular patient compared to control. So the best thing about this particular approach is, even if you have 3 or 4 or 5 samples, you can implement this. But if you have that lower number of samples or condition, in actual scenario, you cannot go with DEGs because DEGs need at least 8 or plus samples. Otherwise, it will give you a very false results because the proper statistical estimation doesn't take place. Because in DEGs we do the log fit, machine learning approach. I'm not going into detail how it works, but there you train model and get the values, and but that is not normalized. So for DEGs you should have a large sample, otherwise it is going to fail and give you very improper result or very less trusted result in terms of significance. So these are the some advantages of this network based approach. So we thought of implementing this approach on the data sets we have. So if you see, we have already implemented this approach on these two paper, where uh, we implemented this on breast cancer and its subtype, and also on triple negative breast cancer specifically. And this work was published last year in iScience. And we have also implemented this particular method on COVID patient cell line as well as patient data to find the actual new drug targets or sort of prognostic factors or biomarkers. And this is also published in Frontiers in Immunology. So today I will be discussing this particular paper because this is more related to cancer and recently published last year only. So breast cancer, as you all know, that breast cancer is one of the leading cancers in the worldwide. And in India, almost if you look, out of all the cancers, you'll find that 60 to 70% of cases are the breast cancer. And the among those breast cancers also, triple negative breast cancer is the most aggressive form, and problematic because we don't have a very targeted kind of treatment, because it is there is no expression of hormones or receptors. So we have 4 different kind of subtypes for the breast cancer. It is basal, HER2, luminal A, and luminal B. And this classification is based upon the expression of receptor and other clinical parameters. I'm not going into very much detail. You can find it online. So this is the study workflow. Okay, what we have done. So we use the TCGA database. TCGA database is the Cancer Genome Atlas. It is maintained by NIH. This project was started in 2011, where we have today we have 23 plus different type of cancer, and you have different kind of data. For example, you have genomics data, that is mutation. You have RNA-seq data or transcriptome data, gene expression. You have epigenomics data. You have methylation, K27AC, this kind of data, as well as you have proteomics data. So they have also provided the protein expression from the same sample. So that is the best thing. You have multi-omics data from the same patient, which is very hard to get because it requires a lot of money to get those kind of data. So what TCGA has done, TCGA performed those studies, performed that sequencing, and it created a database called TCGA, and where the data are freely available. So anyone from anywhere in the world can download that kind of data and do the different kind of research. So what we did, we similarly download 1059 samples, breast cancer samples, and we also had 112 control samples, because control is very important to see the difference or the expression of fold change in terms of any kind of omics data. So these 1059, they have different distribution in these categories. For example, I have not mentioned here the number, but I'll tell you. Basal, we had around 80 sample, 150 samples. In HER2, we have 80 samples. Luminal A was the most one. It has 560 samples, and luminal B was having around 280 samples. So this was the distribution of the number of samples. And what we did for, we computed node weight which is required for the software, because it is a network based software, so you have to give them the node weight on which it is going to work further. So we calculated node weight in subtype specific manner. For every subtype we classify the samples, and then we calculated the node weight, and this node weight was calculated for all the 20,000 genes in every patient. Afterwards, we created a matrix, and then that matrix, say for basal subtype, we have one matrix. In that, for every column is your sample and every row is your gene. So that four different type of matrix we had in the end, and then we run the path.ext on that. After running path.ext, we had list of top nets for every sample in every subtype. Okay, so like 1059 samples were there. So in the end, we got list of pathways in 1059 samples in subtype specific manner. Along with this, we also computed the log fold change to compare our results, that whatever we are finding is actually making sense, and we are showing our result in comparison to DSEQ, that how better our approach is compared to differential expression weights. So differential expression was also computed in two different ways. The first was we simply calculated the log fold change among the case and control, and the other way was we run DSEQ2, which is a very commonly and widely used tool for computing differential express genes. And once we had the list of genes from our approach and the list of genes from the differential expression, we perform various kind of analysis to show the performance of these two approaches. So I will discuss all these one by one. And this particular is triple negative breast cancer. So at the same time, just like what we did here, we also obtained the list of breast cancer TNBC samples. TNBC is also subdivided into four categories: BL1, BL2, LAR, and M. M is mesenchymal. So the same thing what we did here was also done for TNBC. So we'll start with this thing first, and then we'll come to TNBC. Okay. So till now, anyone is having any kind of issue or doubt, or if you have any question, just let me know. Does anyone have any question or any clarification, or anything is not clear? Because it is very important as we proceed further. And don't hesitate. Nobody is going to judge you. Just ask question, whatever you have. Okay. So I think nobody has any question. So I'll start.
Continue career. If
Okay.
they need, they can ask.
Sure, sure, sure. So what we did first, we first perform Venn diagram analysis for path.txt and differential express genes. So the thing is, if you go for, if you want to see that, what kind of genes are actually trying to, is regulating, cancer, whether it is origin or its metastasis, we need to find that kind of gene consistently in that subtype in among most of the patients. Then it makes sense, because that is the one which is actually mutated or dysregulated. So if you see this particular one, TTK genes, it was shown in 175 samples out of 177. It means almost in 99% of the patient, TTK and CDC20, these two genes were dysregulated, okay, or and upregulated as well. But if you see the DGs, only 117 patients showed out of 177 as this as a one of the top genes. It means if you see the comparison of the percentage, these genes were altered in 99% of the samples, but this particular gene was only dysregulated in nearly 67% or two-thirds of the patient. And if you see this particular Venn diagram, you will see that you have 88 common genes, but you also have unique genes. Okay, so this also gives you this impression that this particular method is not only capturing the common genes, but also the subtype specific genes, which is very important to study further if you want to study a particular subtype. Okay, so what we did, we got the five different gene set from here, common and unique for each of the four subtype, and likewise for the DEGs. And then we perform the gene enrichment analysis to see what are the biological processes these genes are associated with. So when you go for the analysis, you expect some results. You want to see that what are the processes which are associated with these genes. So first we looked at the common genes. So if you see, they are associated largely with the cell cycle, mitotic nuclear, chromosome segregation, cell cycle. So cell cycle associated processes was common among all the four subtypes, which makes sense because the basic concept of cancer is what? Proliferation, uncontrolled proliferation of the cells, which is the associated with cell cycle. So this was making sense. But the DEGs did not show any significant geoterms. What does that mean? If you go for a process and you find to see that how many genes are overlapping with that process genes, the DGs did not show any significant geoterms. It means those were very random genes and they are not actually capturing any true biological processes. But our method, it captured the one which makes sense. But we also identified the specific one. So in the basal subtype, we saw the neuron differentiation and T cell proliferation. So it is also very much related to different kind of immune and cell related processes, cell cycle related processes. When we look for the repressed genes, we find that the hormone, metabolic, and muscle regulation processes were repressed, which makes sense. Your metabolic processes get repressed, signaling process get repressed. Okay, so this was what we observed here. But in the specific subtype, we obtained that the same metabolic signaling processes and specific signaling process like terpenoid or retinoic acid, this kind of signaling processes were suppressed. So I'm sure I have shown here for one particular subtype, but you can find the results for other subtype in the original paper. Then what we did, we also looked for the uh, common activated molecular function. So if you see this molecular function, so the common genes were associated with the receptor binding and C2C chemokine, the immune and signaling related event molecular function. And for the repressed, we had hormone activity, peptide receptor activity, what I showed last time in the last slide. So after this gene enrichment biological processes, we looked for the mutation. So what we did from the TCG only, we downloaded the mutation information. So in amplification is how many times that gene has amplified, and in inactivating mutation, we selected the ones like indel, that is insertion or deletion, frame shift mutation. So what we did in subtype specific manner, this is basal, this is HER2, luminal A and luminal B. Okay. So what we did, first we look for the activated amplification. As you can see, it is mostly of them is above zero. Okay. It means that these mutation of these top 100 genes are comparatively higher to all the protein, all the 20,000 genes. Likewise, for the repressed, the relative log fold change was lesser than zero. So if it is activated, it should go above zero, and in the negative one, it should go below zero. If you see here, here also the repressed one, they are showing the negative fold change. It means these genes are more repressed compared to the all the 20,000 genes. And likewise for inactivating mutation, we observe the same results where your activated showed above zero and the repressed shows below zero. And this is comparative change, the change in mutation of the 100 genes compared to all the 20,000 genes. And this 100 genes are the genes which we selected for our analysis in PathEXT as well as DEGs. So now coming to DepMap analysis. So DepMap is a database developed by Broad Institute where you have data for every cancer in cell line specific manner. So for in every cell line, what they have done, they have looked for the expression of a given gene if you knock it out via CRISPR knockout technique. So if you're knocking that gene out, and because of that, if the proliferation of the cells stops, it means that gene is not essential. But if you knock that gene out and the cell proliferation goes very high, it means the gene is very essential for the cell survival. Okay, so here I have shown the results for all the four subtype, where blue is pathy which is our approach, and the orange is degs, which is the approach we are comparing with. So if you see, out of four, all most of this blue bars, they are showing a higher trend compared to orange one. And in this, the value ranges from 0 to 1. So any gene which shows value above zero point 0.5 means they are essential for cell survival. And if you knock that gene out, the cell will not survive. So if you see, our genes were mostly associated with the cell survival, which is essential for cancer. Okay. Now, coming to another validation. So, we were trying to do as much as validation to see that our genes make sense. So, what we have done here, this is driver DB version 3. So driver DB is a database which compiles cancer specific list of driver genes, which is actually driving the cancer. And these are experimentally validated genes. So it could be very useful for those people who are working in the area of cancer. So from here, you can get the list of genes which are associated with driving the cancer. So what we did, we downloaded the list of those genes which are associated with breast cancer, because we are working with breast cancer. So we have downloaded the list of those genes, and then what we did, we performed a overlap analysis. We wanted to see that, say if it is 500, list of genes is 500, how many of our gene and DGs were actually matching with 500. So we call it overlap. So out of, and then what we do Fisher test and we compute odds ratio to show the statistical significance. So if the value of odd ratio is above 1, it means it is significant and is enriched. And if it is below 1, it means it is depleted, not enriched. And this red color shows significant, and the green dot shows non-significance, which is p value less than 0.05. So if you see, for basal, HER2, luminal A and luminal B, all the four subtypes, we compared the odds ratio as well as the significance for PathEXT and DG. See here, PathEXT red, PathEXT red, red and red. In all the four subtypes, our method shows higher significance with higher odds ratio. But the driver genes by the DEGs, they were non-significant. Okay, so this also showed us that okay, our method is doing best compared to DEGs and is actually capturing those genes which are associated with the driver, which are the driver genes in the breast cancer. Okay, likewise, we have another database called Nest PPI. So this is one of the another database which is very useful for the people who are working in the area of cancer, where you find for every cancer they have collected the list of genes which are in a network. And if you knock out the whole network, the cells dies. So it means that those all genes as a network, they are very important. So what we did for our breast cancer, sorry, we downloaded the network of the breast cancer specific from this database, and we had a list of gene set. And just like what we did here, overlap, computing odds ratio and significance, we perform here also. And here also you see PathEXT is performing best compared to TEG in all the four subtypes. It means here also we found that our genes are much more relevant compared to DEGs. And lastly, what we did, we also downloaded a list of genes, not genes specifically, the cancer drugs. So cancer drug database is a database where you find the list of drugs which are approved for a given cancer type, and these are FDA approved drugs. So for every cancer, you have the list of approved drugs. So we downloaded the list of drugs which are approved for the breast cancer, and then what we did, we got the list of genes which those drugs are targeting from the drug bank. And then also, after having that gene list, we perform the same overlap analysis. And here also, if you see, all the four path ext subtypes in all the four subtypes perform much better than the degs. Okay. So what next? We wanted to see if the same trend is observed in the single cell data also. So what we did, we look for the data set where we can find the single cell data for breast cancer in subtype specific manner. And luckily we found one such data set where we have the uh, single cell data, gene expression data in different cell types from in subtype specific manner. So we have cancer, which is epithelial cell, T cell, myeloid cell, dendritic cell, B cell, fibroblast, endothelial. So fibroblast is, you can say that these are the normal cells, and cancer cell, this is the epithelial cell, and T cell, myeloid, these are the immune cells. So what we say, wanted to see, we wanted to see if the genes which we have identified, are they showing higher expression in the cancer cells compared to normal cells or the immune cells. So what we did, we first computed a expected fraction of the gene expression by computing the z score. So first, we, for every gene we computed a z score which is called expected, and then for our 100 genes we computed the z score which is called observe, and then we took the ratio in the log manner, observe upon expected. So what expectation is, it should be positive. Okay. So what we saw, that in TNBC, HER2 and luminal B, the cancer shows the highest expression. It means our genes were showing the very high expression in the cancer or the tumor cells, but the trend was opposite for the luminal A. So this was the only odd one out, we can say, but we haven't went into depth why, what was the reason behind this. But this could be like for future research we can look into this. But out of four subtypes, three subtypes shows higher expression of our genes in epithelial cell type. So next, it's TNBC, triple negative breast cancer. So triple negative breast cancer is a kind of subtype of a breast cancer. Okay. So triple negative means we have three hormones: progesterone, estrogen, and HER2 HER2 receptor. So these three receptor, if these three hormones receptors are absent on a given cell, then it is called triple negative breast cancer. So it is around 15% of the invasive breast cancer. It is more likely to occur in less than 40 years. The survival rate is 91.2 if it is localized, but if it metastasizes, it reduces to 12.2, which is a very low survival rate. And currently the standard treatment includes surgery, chemotherapy, and radiotherapy, but these kind of treatments have a very side effect, so that's why there is a need for a more targeted therapy, or some neoantigen based or a vaccine which could treat triple negative breast cancer. So what we did, just like what we did in the initial breast cancer, for the all the four subtypes, we identified the top nets by running PathEXT. We also identified the differentially expressed gene in each subtype, and then we performed the same analysis like the we performed above. Here also we got some common genes and some specific genes, and then we perform the gene enrichment processes or this kind of analysis. So with the common activated gene, we find mixed kind of processes, which is proliferation, signaling, ion related, morphogenesis, that is development, which is some things are associated, but some were like very new, which could be investigated further. And then we also look for the subtype specific processes. So we got the apoptotic base processes in BL1. In BL2, we have more migration based, where your cells are chemotaxis, showing the chemotaxis movement. The third was LAR, which is more metabolic based, and last M, where we have cell cycle related. So if you see all the four subtypes, they are showing different kind of processes, which makes sense, and that's the thing. And based on this results, we can target or develop subtype specific, uh, therapeutics. So what we did next, we wanted to see if these few genes, they can be used for early diagnosis and prognosis of the triple negative breast cancer. So what we did, from all the 100 genes, we developed machine learning model in gene specific manner, combining all the 100 genes and then selecting feature based. So first we developed 100 different models for 100 genes individual, and then we developed one model where we combined all the 100 genes, and then we did feature selection. Feature selection means you want to see if one gene is, I mean, you just do the clustering, understand, in this manner. And after clustering, from every cluster you are selecting one representative gene. So it means you are removing the redundant genes which are behaving in the same manner. So what we found out, after doing the feature selection, we were landed with 13 genes which were associated with the responder and non-responders. Okay, so what are these responder and non-responder? So we obtained a data set where triple negative breast cancer patients, they have been treated with neoadjuvant doxorubicin or cyclophosphamide, followed by ixabepilone or paclitaxel treatment. Okay, so this is the common treatment which I mentioned earlier, given to TNBC patients. And what happens, we do a follow up study, we look for the how the patient is responding to that treatment. So after a certain point of time, if the cancer relapse, it means if it comes again, it means they are not very much responding to that. But if for a period of time, say 2 to 5 years, if the cancer relapse doesn't take place, means the patient is responding to that treatment. So the patient who is responding is called responder, and the patient who is not responding is called not responder. So what we wanted to see, we wanted to see if our genes can classify these two patients categories. So how this is going to help? So this will help in a manner, say suppose tomorrow if a patient comes to you and say I want to get treated, so what you will do, you just simply do the gene expression profiling of this 13 genes, and you see if their expression is very high or low. If it is, then you can say okay, this patient can respond to this particular treatment. Otherwise, if it is not the same thing, then you can say okay, this patient needs some different kind of treatment or tailored approach. So this helps in precision medicine, what we call as personalized medicine. So this will help you in stratifying patients and giving a better treatment which is going to suit that patient, and irrespective of giving similar treatment to everyone and wasting time and money as well as the health of a patient. So what we did, we used this 13 genes and we developed a model, machine learning model, and you can see with AUROC of 0.71, the model classified the responder and non-responder. The similar thing was done with the DGs also. So these are the 13 DGs which came as the output after feature selection. And if you see, the value of AUROC is lower compared to this. It means these genes are making more sense by our method compared to these genes. So what we found out in the end, that our approach is much better and superior to DEG based approach, because we were able to capture the common as well as subtype specific biological processes, and many of those processes are reported in the original paper, and we have shown that how they make sense, and we have provided enough literature evidence and support where people individually have found those processes in those particular subtypes. And next, we look for the different kind of mutation pattern. That pattern which our genes were showing were more correct compared to DGs, which were like very mixed pattern. Next, we look for the different kind of uh, benchmarking data sets, as I mentioned about the driver gene, the nest PPI network, the drugs, and the genes they target. And there also we showed that our approach is much better compared to DGs. And we also look at the depth map data, that is the cell survival probability of a cell. And then we went to the subtype specific in a triple negative specifically, and we found that in triple negative also the method was able to differentiate in subtype specific manner BL1, BL2, LAR, and M. And also the method is able to capture and prioritize 13 genes which can predict whether a patient is going to respond to this particular treatment or not. So overall, the overall these results signified the importance of our network-based approach. And the best thing is this approach can be implemented to any condition where you have two data set, control and case. So it could be like, patient affected with any other kind of cancer, or diabetic, non-diabetic, or Parkinson and control, Alzheimer patient control. So this is like very general method which can be implemented to any of the disease condition where you have two different sets. And the best thing is it is network based, so it actually utilizes the network which is experimentally validated from the human, is to have it, and the human also. So it provides you the actual reference and not like hypothetic. And likewise, this kind of network has been used by string and other kind of databases where you see the protein protein interaction network. So these are the two projects which I mentioned in the starting. The breast cancer, you can find the code available here, you can download it, it's freely available. And this is the similar code which we developed for the COVID-19. So I'll just give you a brief hint about what we did in this COVID-19. Here also we got the cell line data, where we have cell lines infected with COVID virus and the control. And then we look for the genes which were associated with the COVID patients and the cell lines compared to control. And there also we reported six such kind of genes which are associated with the pathogenicity in COVID compared to control. And not only in that, we also did it in subtype, age, gender, and age diversity based manner, that females are more protected compared to male, younger people are less infected compared to older. So, and ICU and non ICU, that people who have expression of certain kind of genes, they are more prone to go into ICU or develop severe symptoms compared to a group which is having different kind of expression, and they are not that much infected, so they do not generally go to no, ICU. So like this, we have reported that and published that finding in Frontiers, and the code is available here, and the data sets whatever we have used is also available at this particular folder. So thank you, and I will happy to take any question.